Hello students, I am Professor Kumbar AS. Today we are going to discuss about the another theoretical topic known as components or tools of IPM. So let us see what are the tools of IPM. Here first one, monitoring, cultural control, biorational, regulatory control, resistant varieties, mechanical control, biological control and chemical control. So let us see one by one. Here, first component or tool of IPM is monitoring. As we know that monitoring is nothing but keep track of pests and their potential damage. That means just we have to do the continuous observation on the movement of pest or presence or absence of pest and how they cause the damage. That provides the identification of presence of pest, and if you able to identify the pest on which crop which pest is occurred, then that knowledge provides the current pest and crop situation, along with we can decide which exact control method is used or the which exact combination of management strategy is used. So this is one of the most important tools of IPM. Here, as we see that here we can identify the symptoms, or we can identify the disease on the basis of symptoms by continuous observation, and then we can decide which exact control measure is used for that after monitoring. Next tool is resistant variety. As we know that if you use the resistant variety, obviously our yield of particular crop is increased near about 10 to 20%. If we use resistant variety, so that avoids the use of pesticides, they avoids the use of money, they avoids the use of energy, they avoids the use of labors. So that all input or the cost which is required for all that inputs, so that cost is reduced due to the utilization of resistant variety. For example, we can use or that resistant variety is differ from the different crop. For example, HD2109 is a resistant variety against rust of wheat. Then second example is BT cotton. As we know that BT, that is Bacillus thuringiensis. If we use the BT cotton for the sowing, then they avoids or that such a cotton shows the resistance against the bollworm. That is in Marathi it is called as bond ali. That protect or that cotton protect from the bollworm complex. So this is about a resistant variety. The next part of resistant variety is if you do the breeding practices for the pest resistance. That is, as we know that breeding means the crossing. If you cross one resistant variety against the different pest, against or into, if you cross the high yielding variety of different crop, so after crossing such a resistant variety and high yielding variety, so after several generation we can obtain a variety which is resistance as well as high yielding, but it requires several practices and several years. After that, as we know that we discuss about cultural practices. So what are the cultural practices, what is the role of cultural practices? So if you cultural practices, so that make a crop environment less susceptible to pest, and that cultural practices involves deploying burning of crop residues, soil solarization, planting of trap crop, intercropping, crop rotation, tillage practices, use of pest-free seed and planting material, removal of alternate and collateral host, time of sowing, depth of sowing, irrigation facilities, fallowing of the land, use of resistant varieties, field sanitation. So all are called as an cultural practices that helps to reduce the incidence of pests or diseases. So let us see out of them few we have to see one by one. So out of that, first is crop rotation. So what is mean by crop rotation? As we know that crop rotation is nothing but just we have to grow the different crops on the same piece of the land year after year. So why we do the crop rotation? If you rotate the crop in a season, then your fertility of the soil is maintained, productivity of the soil is increased. If you do the crop rotation with leguminous crop, then our beneficial microflora in the soil is increases. That is, beneficial microorganism in the soil is increases, along with better utilization of soil moisture nutrient it also takes place. Then if you do the crop rotation, then there is a less incidence of the pest and disease if you grow the non-host crop in intercropping as well as in crop rotation. Then some of the crop during their growth it secrete toxic metabolites, and that toxic metabolites we have to that toxic metabolites which is secreted by the roots. So those soil-borne pathogen comes along comes in contact with that such a toxic metabolites, so that harmful pathogen is killed. So that crop rotation is also helps to reduce the soil borne population. So there are different number of benefits of such a crop rotation. So this is most important role, that is separation of soil borne diseases. Next, the depth of sowing. So as we know that depending on the size of seeds we have to use the suitable depth. For example, if the here depending on soil conditions also that helps for the depth of sowing. For example, if the soil is warm, so in if you do the sowing in warm soil, so it enhance the seed germination. And if you sow the seed with fully saturated soil with moisture, so germination of plant is takes place but not at that point. That is, germination is slightly inhibited due to the attack of some soil borne microflora due to the presence of sufficient moisture in soil. So depth of sowing is also plays one of the important role for management of soil borne diseases. Okay, for example, again if you take the example, potato seeds planted at a depth greater than 1.5 inch, that is 3.7 millimeter, take a longer duration to germinate and are at greater risk for the infestation of soil borne pathogens, that is wire worms. So that means if you sow the potato seeds below 1.5 inch, so it is beneficial. Then sowing of the seeds, sowing should be done at optimum depth. As we know that depending on the type of the seeds, if you sow the rice seeds, so depth of sowing is 4 to 5 centimeter. For ragi, 2 to 3 centimeter. Sorghum, 3 to 4 centimeter. Cotton, maize, groundnut, 5 to 7 centimeter. That means depending on the size of seeds, depth of sowing is differs. That helps for the better germination, and that helps for less soil borne attack. The next, what about the irrigation facilities? As we all know that in agronomy, in soil science, we discuss about what is the role of irrigation in control of soil borne pathogen. So as we know that if you or if we use suitable methods of irrigation, so we can manipulate, we can manage the different problems regarding weed control, regarding control of soil borne pathogen, regarding us regarding the avoidance of high density plantation, we can avoid several problems. So let us say in first picture, as we know that if we use a flood irrigation, then there is a loss of water, along with there is a growing of a different kind of unwanted weeds also, and there is a loss of water, even though the saturation of even though crop is saturated with a sufficient amount of moisture, even though they cause a heavy loss of water, along with a growth of different types of weeds, unwanted weeds. Then if you use the drip irrigation system, so this is the best method of irrigation system that helps for the better utilization of only use or better utilization of water. They provide a water drop by drop to the plants, so there is no use of unwanted weeds, there is no use of there is no growth of unwanted soil borne harmful soil borne microorganisms due to sufficient soil moisture around the root zone. Then in case of that sprinkler irrigation, so it also useful mostly in hilly areas. So if you use the sprinkler irrigation system in hilly areas, so it is beneficial for the crop, but if there is an attack of pest or disease, so within a short period of time it rapidly spread because this spraying is done continuously or here, so it will be chances for the spread of disease due to the sprinkler irrigation. So out of that flood irrigation, then sprinkler irrigation, drip irrigation is most useful for the controlling of pest pathogen. Next, intercropping, as we previously discussed in crop rotation. If you do the intercrop, for example, if you do the here tomato will be there, and here if you do the intercrop with the marigold, so it is beneficial to reduce a pest population, because that marigold, the roots of marigold, during their growth, it secrete a alpha terphenyl, is a poisonous alkaloids, and that alpha terphenyl kills the growth of other harmful pathogen without cause damage to the beneficial pathogen which is present in soil. Then that intercropping provides the better utilization of land. If even though there is a failure of main crop due to the any natural calamities, so that crop gives a some economic yield. Then it when we do the intercropping, so it does not require any special practices. So these are the use of, or these are the role of intercropping in plant disease management. Next, as we know that here deep plowing or burning of crop residue. So what is the role of deep plowing? As we know that if you do the deep plowing, so whatever the pupal stage or nymph stage or larval stage or the eggs of different insects what that are present inside the soil, so due to deploying that are comes outside, what are the larvae or the eggs or the pupa that are exposed to outside or on the surface of soil, and that are exposed directly to the sun during a afternoon session, and when they expose directly to the sun, so ultimately the larval or pupal stage or eggs of different insect are completely killed. So in that way, deploying is beneficial for controlling the pest population or disease. Then next one is burning of crop residues. As we know that most of the time in wheat or in sugarcane, after the harvesting of the crop, whatever the trash is remaining, so that trash is or crop residue is completely destroyed. It having both beneficial and harmful effect. What is the beneficial effect? During that burning, most of the harmful pathogen is killed. This is beneficial effect, but at the same time, whatever the natural enemies or natural biocontrol agent is present, or beneficial microorganism is present in crop residues or inside the soil, so due to the heating, that are completely killed. So these are harm, or these are disadvantage of that burning of crop residue. Next, use of trap crop. As we know that previously we discussed about the in intercropping. In case of trap crop, so we can show trap crop is nothing but it is an crop which are planted that are attract to the number of agricultural pests or diseases to avoid the main crop, to avoid the attack of main crop. That non-host crop is planted in between the rows of two main crops. So that trap crop attract the different types of the uh, agricultural pest, different kinds of insects, and due to that our main crop is prevented. So this is about your trap crop. As we discuss about uh, trap crop, for example, if you use the marigold around the crop, so that marigold kills the different soil borne pathogen due to the presence of alpha terphenyl, which is secreted by the roots of marigold. The next, different types of sucking or different types of insect that are attracted towards the flower of marigold, and due to that our main crop is prevented as it is. So that are the role of trap crop, to, to trap the pathogen or pest which is harmful to main crop. The next one is soil solarization. So soil solarization, as we know that the soil is covered with the, before the planting, the soil is covered with the polythene sheet, black colored polythene sheet. So due to that soil solarization, particularly in summer season, due to high intensity of sunlight, that polythene sheet is get heated, and due to that heat conduction, whatever the soil borne pathogen which is present inside the soil, due to the conduction of heat, that harmful soil-borne pathogen is completely killed. Near about 30 to 40% soil-borne pathogen is killed due to the soil solarization. For example, Fusarium, Rhizoctonia, Pythium, Phytophthora, such a soil-borne pathogen is killed due to soil solarization. Next one is trap cropping arrangement. As we discussed in previously, for example, here this is cotton, this is your main crop. Cotton, this is your main crop, and in between that cotton, if you plant alfalfa, is a trap crop. So alfalfa is a trap crop that attract a different kind of the pest, and due to that only trap crop is infested with the different kind of the pest, and our main crop is remain as it is. There is a less incidence of pest on a main crop, or our main crop is prevented from the incidence of pest or disease. The next one is flooding. As we know that flooding is not beneficial, as we discussed in previous lecture, but the another uh, positive point of flooding in case of rice is there. Flooding is followed to control the problem of soil borne pathogens. For in case of that, we reduce the weed, or we can reduce the number of fungal propagules, that is number of harmful fungal pathogen or insect and nematodes which is present in soil. For example, Phytophthora, Rhizoctonia, Fusarium, Verticillium, Armillaria. So these are called as an important soil borne pathogen which is present in soil. But due to flooding, anaerobic condition is formed. That is, there is a scarcity of oxygen in soil, and due to the unavailability of oxygen in soil, most of that harmful pathogen is completely killed. So not all, but in some cases, like in paddy, we can use a flooding system. Next cultural practice, that is called as an fallowing of land. In Marathi it is called as padik jamin ठेवणे. Sometime what happens, after plowing we can keep our land as it is without any cultivation, as it is. So if you keep your land as it is after plowing, so if you keep as it is for a one season or two season as it is without any cultivation, if you keep your land as it is, that is fallow, without cultivation, then your soil is enriched with a beneficial microflora. Then your soil is enriched with optimum soil moisture. Then most of the soil borne pathogen is completely killed because there is no any crop in our field, and due to that, whatever the food is required for the harmful microorganism which is present in soil, that are not that are not present at that situation. So the incidence of pest and disease also less. Soil is completely enriched with different kinds of micronutrients, and soil water holding capacities also increases. So for that purpose, in most of the places, that felling of the land is that practice is followed. Next, here the field sanitation. As we know that why the field sanitation is essential, because most of the pathogen is present in plant debris. That is, after the harvesting of the plant, whatever the plant part is remaining, that is plant debris, that are remaining as it is. And when the favorable condition is arises, then they directly attack on the main crop. So if you clean your field by collection and destruction of plant debris, so pathogen do not get a proper place to complete their life cycle, and due to that pathogen get automatically killed. So that simple method is there, that is field sanitation. That is infected part of the plant, we have to collect and destroy it from the field also. Next, one most important part is there, use of pest free seed and planting material. So as we know that most of the plant diseases is spread due to the use of diseased or pest affected planting material. So better way, we have to use a tissue culture plantlet. For example, in case of sugarcane, we have to use plantlets, or we have to use the tissue culture uh, planting material in case of sugarcane to avoid the spread of diseases. Next one is there, last one, that is removal and alternate of collateral host. So as we know that, first of all we discuss about what is the concept of alternate host and collateral host. So alternate host is nothing but, as we discussed in previously, the pathogen which complete their life cycle on a main host. For example, here main host is wheat. On main host, that is main plant, Pakshinia survive, that is rust, Pakshinia fungus that survive on the wheat crop. But in absence of wheat or after the harvesting of the wheat, that pathogen, that is Pakshinia fungus, that complete their life cycle on the another host, that is weed. For example, weed. For example, barberry weed. Barberry is a name of weed. On that weed, that Pakshinia graminis, that is fungal pathogen of wheat rust, that fungal pathogen complete their life cycle on barberry weed in absence of wheat, that is main host. So if you remove the alternate host, that is barberry plant, if you completely destroyed the such a weed, then there is a no place for the Pakshinia to survive their life cycle in absence of main host. Then in such a condition, due to unavailability of food material or the place, Pakshinia get killed. So that it is necessary to remove the alternate host or collateral host. So alternate and collateral host means what? It may be a weed, it may be any noxious plants on which pathogen complete their life cycle in absence of main host. So it is very essential to remove the alternate and collateral host. So what is the alternate and collateral host? That is, those pathogen complete their life cycle on weed or any other crop residues in absence of main host. So this is called as your alternate or collateral host. So this is about all the cultural practices. In this is our first part of our tools and components of IPM. Then second part we have to discuss in next lecture. So if any difficulty in that topic, so please ask me. Thank you.